Okay. Okay, so I'm going first today. Many of you know me, I'm Sandy Eckel. And here's just a quick sort of overview of the sorts of things that I do. So I really enjoy teaching. Many or some of you are in my 511C class right now in the fall. That's the correlated data analysis class. In the spring times, I also teach a newer class on data science for climate change and health. I'll talk about that in a little bit. I also direct our PhD program. And my research generally is highly collaborative. I work a lot with folks, especially in environmental health. And I've listed for you here a couple of the different collaborations I have. And then the things in blue are things that I'll mention in our speed talk. But increasingly, my research is really focused on climate change and health. And so I'll be talking about a number of areas of research there, in particular, pilot work that I've done with a PhD student actually in PIBS, and a project with Dr. Erica Garcia on electric vehicle adoption. And then also, I don't have any official news yet, but we're hopeful that a P20 center will be funded related to climate change and health. Rima Habra is the overall lead, and Erica Garcia and I will be project leads of one of the research projects. And then I also have work on decarbonizing medicine, but I'm not going to talk about that today. I'm interested in climate change and health, especially these days, and climate change is a really hard topic to be thinking about. And increasingly, we have real messages about the urgency of acting on climate change and health. You see messaging like this where we have, you can see my cursor, we have like really decision points where we have to start making changes like right away. And so I find this topic can be very overwhelming, even though one might want to get involved in it. And so I've been trying to think about ways to get involved and ways to think about this area of research. And I would really, last year, a couple of years ago, I found this way of messaging and thinking about climate change and health. This was from the Lancet Commission in 2015. They were talking about how climate change is the greatest global health threat facing the world in the 21st century, but it's also the greatest opportunity to redefine the social and environmental determinants of health. There's going to be major changes coming, and if we make these changes in smart and effective ways, we can have really big impacts on increasing our health overall, hopefully. There's a really great video you can watch. I don't have time for right now, but you can look at it if you want. So the key takeaway is that climate change is a really great opportunity for us to be furthering public health. So I'm going to talk about two projects related to using biostatistics to try to drive forward climate and health solutions. The first is a little bit more methodological, and the second is a little bit more applied. The theme being, the first one being evaluating and improving methods for how we can quantify the association between a climate hazard and a health outcome. The second one being a little bit more solutions focused. How can we drive forward, solutions that by looking at the co-benefits of these mitigation measures? So that first one I'll start with. So this is work that was funded by EHSC Climate Health Pilot Project. And it's work with PIB student Caleb Shimke. Should have asked him how to pronounce his name first. But the title of this pilot project was Extremely Important and Extremely Challenging Methods for Studying Extreme Climate Hazards in Health. The motivating idea here is that climate hazards are often very rare that we're interested in, but increasingly relevant. They happen more frequently, but they're still rare. And here's just a figure from the New York Times a number of years ago. Looking at the distribution of temperatures, and you can see how that extreme upper tail is increasing over time. So in looking at the literature that's out there on quantifying things like relationships between temperature and health, especially the acute impacts, much of the methods that are used have been adapted from air pollution epidemiology approaches. And they really haven't been evaluated for rare exposures, which is the setting in which they're being applied. Some of the common methods that are used are time series studies with quasi-Poisson regression, where the key focus is how do you adjust for smooth functions of time, and also time stratified case crossover designs, where you control for time invariant confounders by design, and then you also control for time by the way you do your reference sampling. This is increasingly popular due to those features. But some concerns methodologically are that there was a really nice paper that was actually using a little bit of an older case crossover design, but that paper showed that case crossover designs are less efficient as compared to time series studies. And then the question that arose in my mind was, is this problem exacerbated when we're looking at a rare exposure, especially when it's been dichotomized? And if we're using a conditional logistic regression approach in a case crossover design, was this problem going to be even worse as we're looking at more and more rare exposures? So the goal of this project was to evaluate the statistical performance of these two methods and relating rare exposures to an outcome like mortality counts and sort of administrative data sets, large data sets. So the idea was looking at we're going to perform a simulation study comparing the two methods. We were going to base the simulation on real observed temperature and mortality trends in LA County. You can see here on the bottom, you can see the daily minimum temperature over about a 6-year period in Los Angeles, and you can see the corresponding daily death counts in Los Angeles County. Also, the highlighted data points there are from the top 1% of high temperature days. And you can see that those happen really just only a couple times a year. Sorry?
What do you mean by highlighted?
Highlighted with the little black triangles. And so you can see various ways that we could model the trends in time. So basically what we did here is we generated data under several simulation studies, data generating scenarios, where we would assume sort of data generation that corresponds to a time series approach or data generation that really corresponds to a case crossover approach. And then we would also have a continuous X or a binary X that was increasingly rare. And basically what we found was that the case crossover design has worse efficiency than time series, like in the previous study, but using the more modern time stratified case crossover, it wasn't quite as bad as the older version of case crossover. But this a decrease in efficiency got worse for more rare exposures as we might expect. And then like a large body of literature, we found that either method was biased if you weren't appropriately controlling for time. So this just brings to light the fact that we really do need methods that are tailored to looking at these rare exposures, because we want to do a good job quantifying the impacts of rare exposures because they're going to be more and more important in the future, but we don't have as much data. Then another angle at which you can start to think about climate change and health is thinking about mitigation measures and how we can evaluate whether measures to mitigate emissions, whether they have benefits in terms of, air pollution, for example, and health. And maybe that can inform policymakers in making these tough decisions about which mitigation measures to encourage. And so for this project, I do have a conflict of interest that I want to disclose. The PI, Dr. Erica Garcia, has some stocks in electric vehicle companies, and I actually have a spouse who works for an automobile manufacturer. So this is a work related to cars, so I have to make this disclosure. So in this project that the PI is Dr. Erica Garcia. This is a project looking at electric vehicle adoption in California. And some of the preliminary data that we published that really helped get the grant was related to looking at a natural experiment in California and seeing how, as within certain zip codes, as the number of electric vehicles registered have gone up over time, is that related within zip code to reductions in air pollution and reductions in asthma emergency department visits. And so this publication, we talked about the evidence that we saw. It got a lot of press at the time. And we also unveiled a really important adoption gap where, communities that have lower levels of education also are really lagging behind an electric vehicle adoption. So any co-benefits that are being experienced, they're potentially missing out on these. So it's a really important issue. This R01 that Erica has is helping to dig into these issues, and we're having community dialogues trying to understand these issues and so on. But one of the things I wanted to bring up here was that this trying to make this link between the number of EVs and air pollution, we started looking at EPA monitor data, and there's about 100 different EPA monitors across California. And so it's a pretty small sample size to try to look within EPA monitor over maybe a 6 or 7 year period during this study. And so we found evidence in the hypothesized direction. You can see here a negative association, but it wasn't statistically significant, really because of reductions in a low powered study design here by having a small number of zip codes, and the zip codes where the EPA monitors are located. It's not randomly selected throughout the state. It is sort of purposefully selected. And so our ongoing work that we have going on right now is trying to actually use satellite measurements of NO2 air pollution. So we get statewide coverage. A downside is that we have less temporal coverage, but this is an area where we're working on right now. You can see that the time period, if you look here, it does include the pandemic. So we are being very careful in thinking about how to appropriately account for the pandemic period. But I think this is going to be a new direction to really help quantify, from space, the impacts of mitigation measures on air pollution. And so this could be the methodologies that we're developing here, the approach that we're developing could be used. California's a great case study because we have a lot of pollution monitors on the ground and we have a lot of EVs, but it could be applied, all over the world in theory in the future. Okay, and then I'm running low on time, so I just want to mention other projects ongoing. I want to give a big shout out to Brittany Marion, a PhD student that I work with. And she, last year she got an F31 award, our first biostat student to get this. And so she's working on interesting methods, looking at ways to characterize multi-pollutant patterns using diurnal patterns within a day and doing unsupervised time series clustering. And so she's thinking about methods to identify the number of clusters because it's a very underdeveloped field, and how to then translate these types of air pollution days into an epidemiology study. And then my very last slide, I just want to give a plug for our class that I've developed with Erica Garcia and Sam Silva on data science for climate change and health. We've offered it twice. This last spring, we had introduced a lab, so now it's a 4-unit class. This fall, we're applying for a proper course number, and then Sam was planning to be on sabbatical this spring, so we're actually going to be teaching it next spring in 2026. So keep on the lookout for that. So that is it. Thank you very much.
There is a question online.
Okay.
So we have the questions or wait at the end?
Yeah, no, please ask questions now. Good.
So I was super sorry, as you were describing this, it reminded me, did you try to do some hyper local kind of pollution information like purple air as opposed to the EPA high quality sensors or?
So we have not, but that's a great idea to incorporate other local sensors. So that's definitely another direction to go.
And then the other question is, I mean, is the reduction with EV use really for EVs, or is it just that the regions that are, the zip codes that are wealthier tend to be better areas, they have lower air pollution to start with, and also higher education and higher income and higher EV adoption? Because I mean, it's quite hard to, just by personal experience, it seems a bit hard to disentangle.
Yeah, we're trying to be very careful about adjusting for these time varying confounders. Like we're including variables like percent work from home, changes in SES at the zip code level and things like that. But it's definitely a challenging issue, especially when it covers the pandemic period where these things change a lot.
Yeah, thank you.
Thank you.
Awesome work.
Okay. Excellent. Thank you. Kelly Street is next. All right. Cool. All right. Awesome. Thanks for having me. I'm Kelly Street. I'm a no longer super new assistant professor in biostats. This is my 3rd year. This is my 2nd time giving one of these speed talks. Last time it was just single cell omics, but now I think I've progressed into single cell multi-omics proper. So that's my new title. Quick plug for my class, Introduction to Health Data Science, PM 566. I think it's a really cool class, teaches a lot of great computational skills for working with data. Check it out. All of our materials are online if you just want to try out a particular thing. Okay, great. So a lot of people have seen plots like this. This is a Manhattan plot. It shows for a given disease or a particular thing that you're interested in studying, how each SNP or change in the DNA of an individual associates with that disease. So if we're looking at, say, lung cancer, these might be spots that are associated with increased risk in lung cancer. That's great. How do you then figure out what parts of the body that disease is affecting? This allows us to determine which individuals might be at higher risk for a disease, but how do we actually understand the mechanisms of disease? That's, I think, the key strength of single cell. So as you're probably aware, your body contains lots of different types of cells. This is the Human Cell Atlas, which has single cell reference data on a lot of different types. They've got blood cells, they've got kidney cells, they've got bone cells, they've got liver cells. You've got a lot of different types of cells in your body. And it's important to be able to disentangle the single cell effects from sort of the bulk effects. When we have bulk data, which is sort of the traditional RNA-seq, or even, when we think about SNP data, changes to the DNA are sort of at the bulk level because they affect, for the most part, your entire body, or at least every cell that comes downstream of the one that has a mutation. When we've got single cell data, we can actually see all the different things that went into that. We can see all the different types of cells, and we can measure changes in them. So I said lung cancer earlier. If we're looking at lung cancer, we can look at the different types of cells in your lung and see how are they behaving in healthy people and how are they behaving in people with this disease. And we can actually start to quantify those differences and potentially find therapeutic targets. So one of the things that I'm super interested in right now and that I've got data sort of coming in on is single cell RNA-seq and single cell attack-seq. So for those of you who aren't familiar, RNA sequencing has been around for a while. You've got your genome, there are genes within your genome. Those genes get transcribed, which means turned into RNA, and then RNA gets turned into proteins, which is how cells do things. We can sequence that messenger RNA. We can put it into some chemistry and a fancy machine and actually count the number of messenger RNAs for each gene in each cell, and count them up and get this big matrix of counts for each gene in each cell. It's not perfect, the capture efficiency rate is not great, but whatever, you can get a pretty good sense for what each cell is doing. ATAC sequencing is a little bit new. Assay for transposase accessible chromatin. It measures which parts of the DNA are open, which parts of the DNA are accessible, and that's an important feature that is thought to be necessary for gene expression. The gene has to be accessible in order for the gene to be expressed, particularly the transcription start site of the gene. So we might get something like this, where we see sort of collections of reads stacking up at various points along the genome. Some of them will be along the transcription start site of a gene. Some of them will be sort of in these open regions. And maybe those correspond to enhancers or promoters or other functional elements where proteins need to bind to DNA. This is a little bit trickier to quantify. We have to use some sort of fancy algorithm called a peak calling algorithm to identify peaks within these, data-driven peaks. And then we quantify based on those peaks. So we count up for each cell, how many reads did we observe in each of these peak regions. And that gives us sort of the second part of this type of data. So altogether, we can do these assays on the same individual cell. And this is a very recent and very exciting development that has only come about in the past couple of years. Before, these were two separate assays. We could only do them on separate sets of cells, but now we can actually do them on the same individual cells. It's really exciting. There's a ton of stuff that can come downstream. This is an extremely rich data type. You can be thinking about dimensionality reduction, more dimensionality reduction for visualization, clustering, cell type identification, differential abundance, differential expression. There's just a ton of work to come downstream of this very large, very rich data set. I think it's a great target for sort of advanced machine learning algorithms. If you want to do deep learning, this is a great place because we've got complex interactions between the genes and the peaks. There's a lot going on in this data. I'm going to highlight one particular aspect, which is the problem of joint embedding. Shinren Wang is looking into this with my group. This is basically how do we create a low dimensional embedding, something like PCA, something like what we would do with a single assay, but do it for multiple assays. How do we somehow summarize the information that we've got in all these different assays? In our case, it's only two, but still, in some low dimensional space where we can do traditional statistics, we can do traditional clustering based on Euclidean distances, things like that. Another interesting problem that I'm looking at is feature mapping. So a lot of people will try to do some sort of mapping between the peaks from the ATAC-seq and the genes from the RNA-seq, and usually they just do it based on which one is closest. And that's not necessarily the best way to do that. So we're looking at using AnnoQ, which is a great tool for annotating variants in the genome and using, leveraging that information about where we see variants in the genome. And we know that those variants then have downstream effects on genes. So can we use that to link peaks to genes in our multi-omic data? Okay. The other thing that I want to talk about quickly, I'm gonna check my time, okay, is spatial transcriptomics, because this is something that I'm really interested in. Lu Zhang and I are currently writing a grant proposal on spatial transcriptomics, and we've got sort of a small working group on this. So I want to talk about this and see, hopefully generate some interest. First of all, spatial biology is, like, in some senses, a new field, and in other senses, very much not, because it can mean a lot of different things. So I'm going to talk about two things in particular. When we talk about spatial transcriptomics, we're usually talking about one of two things. There are sequencing-based assays, like the one on the left, where we have basically a really, highly particular slide that has a bunch of little spots on it. And at each of those spots, we can pull down all of the RNA or all of the DNA, whatever thing we're interested in sequencing. And so basically each of those individual spots becomes like a cell in a single cell data set. It's not, and it can actually contain information from multiple cells that happen to overlap the same spot, but it's kind of similar in terms of how we analyze it. The other way that people do spatial transcriptomics is with imaging. And this would be something like imaging mass cytometry, and I'm not going to talk about this a whole lot, but this is also a data type that I've encountered where it's all based on microscopy. It's based on fluorescence. And usually you can only do a panel of genes this way, or a panel of proteins, but you can get extremely high resolution information about where genes are being expressed or where proteins exist within the context of a larger sample. This has a whole bunch of different challenges that it's a whole rabbit hole I don't want to get into, but it's really cool data to work with. The data set that I'm most excited about right now is actually a spatial integration problem, where we've got two data sets of that imaging type that I mentioned before, where we've got 246 genes by hundreds of thousands of cells. Like, these are large data sets in terms of the number of cells, but pretty limited in terms of the number of genes. And then we've also got, from the same system, this is all from kidney, traditional single cell RNA sequencing, where we've got all of the genes, but a slightly more limited number of cells. Just to give you some cool pictures, this is sort of what that looks like. Those are those two spatial data sets that I mentioned, just colored by a couple marker genes. And here's a UMAP plot, which is a nonlinear embedding method. You can think of it like PCA, but better for making pictures, worse for doing downstream analysis. This is just a visual representation of those data sets. And there are ways to basically do spatial integration. How do we use those 246 genes where we have information across all three data sets to create some sort of joint embedding? And in this case, you can clearly tell that that embedding is based on the single cell RNA-seq. That's the same UMAP plot that I showed before, but we can actually map all of the spots, or in this case cells, because it's imaging based, onto that UMAP embedding. And if you look closely, you can see that it sort of fills in some of the gaps here, which is actually a bad thing, because it means we're getting cells in places that sort of fall between different populations of cells, which is probably, a reflection of our limitations in sort of identifying cell types within the spatial data. The problem that my collaborator is interested in solving, which I don't know that we can actually do, Lou and I have both sat down and thought about this, and we're like, I don't think this is possible, is imputing the expression of all of those, 32,500 missing genes that aren't in the spatial data. Trying to impute the expression of a single gene in a single cell, extremely difficult. I don't know if it can be done. But yeah, this is my best attempt. It's not very good. Okay, I believe that's all I've got. So thank you very much.
Any questions for Kelly? Okay.
Me and Dr. Conti have talked about kind of UMAP and Disney stuff
Yeah.
in the past. Now, I was just wondering if you had any thoughts on the differences in the methodology between UMAP and Disney, specifically when it comes to single cell analysis.
Yeah, that's a great question. TSNE was like the original nonlinear dimensionality reduction. And they both are sort of designed to show cluster structure in the data. TSNE in particular is only interested in local structure. The difference between the two is just the distance metric that they use for the embedding. So they both share that principle that I said, where they're better for visualization, but you shouldn't be building downstream analysis on top of them, like either one. But UMAP is a little bit more tailored for preserving some of the more global information. And so, you know, in TSNE, if two cell types are on opposite ends of the plot, that doesn't mean anything, because large distances don't mean anything, only local distances. In UMAP, they try to balance that out a little bit more.
Just, so Natalie will be next, but I'll just add, you say better for visualization. Typically better for visualization for showing clusters.
Yes.
All of us got in a lot of trouble by underrepresenting the variation in human genomic, ancestry by using a UMAP as opposed to principal components or something like that.
Yeah.
It shows the clustering, but it underrepresents the variation.
Yep.
Yeah.
Yeah, I was wondering, so you have a lot of cells, but a much smaller number of cell types, presumably.
Yes, yeah.
And so with the open chromatin analysis, with those cells that are the same types of cells, how much variation do you find compared to the different cell types in the open chromatin regions?
That's a really good question. In my experience, you see, sorry, what I say is, it's easier to distinguish cell types based on RNA than based on ATAC sequencing. You can still do it, and you can even use like known marker genes to do it, because some of the peaks do overlap the transcription start site for specific genes. And so it's still possible to like do your clustering on a TAC-seq and look at averages within a cluster of particular marker genes and say, okay, these are cell type 1, these are cell type 2. It's just a little bit harder in a TAC-seq. One of the things I'm actually currently looking at is in the upstream pre-processing, how to do the peak calling matters a lot. Most people do the peak calling per sample, which means they're throwing a bunch of different cell types together and doing the peak calling. But actually, if you do the peak calling by cell type, then you might get more informative peaks that are sort of specific to each cell type.
Yeah, so that makes sense, that you essentially would assume that they're gonna be pretty similar within the cell type.
Yeah, yeah.
Okay, great. Thank you very much. All right, next is Natalie Delrocco, and she is joining us via Zoom. Thank you. Oops, we're having trouble hearing you. Hold on. You are off.
How about now?
Yes, now we can, yes.
Excellent.
Perfect.
Okay, sorry about that. Hi, everyone. Thanks, Dave, for the introduction. And you can see my screen okay, right?
Yes.
Fantastic. Okay, so my research is a little bit different than what we've seen from Kelly and Sandy, which is kind of typical, but I'll go ahead and get started. Let's see. Just a little background, since I do work remotely, I don't get to see all of you very often, but I'm a recent graduate from the Biostatistics program at the University of Florida. I've done prior research in pain and acute lymphoblastic leukemia, which is a type of pediatric cancer. And I'm a new-ish, I just had my first one-year anniversary assistant professor in our division. My clinical research interests are osteosarcoma and Ewing sarcoma with the Children's Oncology Group. And my methodological interests, I have done some outcome-dependent sampling previously. I am always looking into statistical considerations for clinical trial design and analysis by nature of working with the Children's Oncology Group. And today I'm going to run a project by everyone related to clinical prediction models, which is near and dear to my heart. It was part of my dissertation, and so I'm excited to continue. The project overview, the working title right now is Publishing Prediction Models in Clinical Oncology, a summary of guidelines and recommendations. And so where this was born from was being involved with the Children's Oncology Group. I have gotten to see the editorial side of publishing research in the field of clinical oncology. And these days, that is very entangled with the field of clinical prediction models. So as I said, many original research articles in clinical oncology right now feature an element that is clinical prediction models. And the problem that we're seeing on the editorial side is that there is a really heterogeneous background of research teams in clinical oncology. So there's almost always some sort of quantitative researcher on the team who is working on a project, but that can range from a statistician, like most of us are in this department, or it could be a quantitative geneticist, an epidemiologist, a mathematician. It really runs the gamut. And the reason that that is challenging is because statisticians often know how to properly report a model for publication so that it can be evaluated and determined how useful it's going to be in clinical practice. That's not true for every heterogeneous quantitative field. There are tons of fantastic guidelines that are out there and available. In particular, I want to highlight the Equator Network. So if you write guidelines for proper reporting of a specific quantitative analysis or a specific study, you can actually get them certified with the Equator Network, and they will appear online for people to reference so that things can be properly published. So the challenge with that, as great as Equator is, the guidelines are disjointed and they can be really inaccessible if you don't come from that statistical background, particularly for clinical prediction models. So for example, many clinical prediction models are prognostic in nature. So you're trying to predict a patient's outcome at using baseline factors. And if you look at guidelines for diagnostic and prognostic models in Equator's search base, there are 32 sets of guidelines that pops up. And that's just really, it's not very practical to ask researchers to go through 32 sets of guidelines and make sure that they are following each set of guidelines for a quality research project. In addition, even if they can find the guidelines that are correct for them to follow, some of those guidelines can be inaccessible. They're really written for statisticians or geneticists or mathematical modelers, depending on the type of project that you're doing. So the purpose of this project is to really, for clinical oncology research, condense what guidelines researchers should be following and make some recommendations for commonly missed elements that lead to a paper not being able to be evaluated as quality or not quality for publication. And as I mentioned, it's a big barrier to the dissemination of quality research right now. We're having to not accept a lot of papers due to missing elements that can be captured if the proper guidelines are followed. And I think a lot of the confusion comes from just what is a clinical prediction model? If you didn't get to spend your whole dissertation thinking about clinical prediction models, it might be a foreign term, but it's really just any model-based attempt to predict clinical outcomes based on a combination of patient characteristics. And so that runs from very simple to very complex. There are examples where researchers take a simple sum score, assigning 1 point per prognostic factor, and add them up to see how risky a patient might be, and they'll correlate this with a clinical outcome. Traditionally, what I and probably most people in the division would think of would be a multivariable regression model, typically for a survival outcome, but it also gets as complex. And these days, this is where we're running into big challenges. It can be neural networks for survival outcomes that incorporate thousands and thousands of factors. And again, this is increasingly represented by machine learning and artificial intelligence. And we'll circle back to this in a bit. So our purpose here is to, again, really condense for people what guidelines should you be following out of the hundreds that are available. And we're proposing that this doesn't have to be terribly complicated. The first step is to determine your data source. If your data source is coming from a clinical trial, you can follow the consort reporting guidelines. If your data source is coming from an observational study, you can follow the Strobe reporting guidelines. But the really important thing is, no matter what kind of study you're putting out there, whether it be genetics, whether it be a regular regression-based study, or whether there's a more complicated machine learning style analysis, you have to describe your data source and how your data is coming into your final model. Otherwise, there's no way for an editorial board to understand how applicable your analysis is going to be to cancer populations moving forward. And then for analysis reporting, there are really 3 sets of guidelines that we are urging authors to focus on. So REMARK, which is reporting recommendations for tumor marker prognostic studies. So in clinical oncology, many of these studies are incorporating a novel biomarker or a novel genetic factor. And following the REMARK guidelines there, make sure that you are reporting everything that is needed to make sure that that new biomarker has been properly validated. And then the tripod guidelines, which are probably most relevant to this group, these are specifically for multivariable prediction models for prognosis or diagnosis. And they have 2 sets, regular tripod for models that maybe we would typically use, and then tripod plus AI. And this is just an extension of tripod for incorporation of machine learning methods. And I think I'm doing okay on time. I have a couple more minutes. Okay. And so we're proposing that after you determine where your data can come from, it really is, for most studies, as simple as this. Do you have a new biomarker that you're incorporating? And are you incorporating some sort of machine learning or artificial intelligence? If the answer is yes to both, take a look and follow REMARK and tripod plus AI. If you are incorporating machine learning or artificial intelligence, but you don't have a new biomarker, just focus on tripod plus AI. If you have a new biomarker, but are not incorporating machine learning or AI, you can look at REMARK with regular tripod. And then if neither is the case, we can follow regular tripod. And this is going to cover about 90% of the research studies that we're seeing come in. If they follow these guidelines, they are going to have, from the get-go, they're going to have the elements that our editorial board needs to decide whether the study is quality and can be applied in the future or not. Of course, there are the rare, case control studies or things that may not fit into these guidelines. But the goal here really is to make sure that most of the original research coming in is equipped, and it's much easier to do that if the researchers are following guidelines from the get-go, rather than getting the comments from reviewers down the line. So in conclusion, we are summarizing and simplifying the guidelines related to publishing a high-quality clinical prediction model in clinical oncology. I'm not getting into this now because it's quite weedy and I don't have much time, but we are providing recommendations related to statistical analysis of traditional clinical prediction models. So, for example, oftentimes investigators don't consider what missing data method they should be using and just rely on complete case. or they dichotomize a continuous factor without also presenting an analysis of what the factor would look like if they left it as continuous and modeled it with a spline, for example. So providing little recommendations where they can make their research a little higher quality to meet that publication bar. And this is sort of a next step and really my ulterior motive for presenting today. We would like to do the same thing, providing recommendations related to the analysis of machine learning and artificial intelligence-based clinical prediction models. That is not my field of expertise, and it's actually there's a big, it's sort of a gap in editing where in clinical oncology, where we are frantically searching for journal editors that have experience in machine learning and artificial intelligence-based prediction. So if that's something that is interesting to you and you have experience with, I would very much love to collaborate with you. References, and thank you.
Thank you very much. Any questions for Natalie? Don't see any in the room. So thank you, Natalie, for that.
So actually, I have one question. It's not too late.
Yeah.
So what is the major difference between tripod and tripod AI? Like, how does the AI component change the basic set of guidelines?
So it mostly has to do with reporting of hyperparameters and the validation that you're using to set those hyperparameters. So for example, if you're using cross-validation or bootstrap or something to define those hyperparameters, you have to fully describe that so that, you know, an editorial board could judge whether that's sufficient. So those slightly the more complex elements of a machine learning model that you wouldn't find in a traditional statistical model, that's where that expands. But I think that's actually a really good point. There isn't much difference, but the people who are putting machine learning models out don't meet the basic reporting requirements of a regular statistical model. So that's also something we're trying to raise awareness for. Even though you're not doing a regular statistical model, you're doing a machine learning model, you still have to report that fully and properly like we would with a traditional statistical analysis. But yeah, it's not much different. It's a small extension.
Thanks.
Thank you. Wait, one more question?
This is coming from maybe not as heavy in the biostats of machine learning, but I've seen in certain like online lectures and stuff where places will consider regressions as part of machine learning at times. Is that when you're saying like a simple statistical model, in this case, would a regression be considered a simple statistical model or part of a machine learning?
That's a great question. I guess in my line of research, I have always considered a regression model, like a traditional statistical regression model, to not be considered machine learning. I think there's some buzz there because, you know, maybe as a field, we don't have a great and perfect definition of what is considered machine learning or not. But a regular regression model, I would not classify as needing to use the tripod plus AI guidelines, just regular tripod.
Thank you.
Thanks.
All right. Thanks, Natalie. All right, we'll go. We're not going to make the 12:50 cutoff. So if you do have that class, feel free to leave now. Chun, where is your?
I don't know. He called it something.
Oh, what's it called?
2024 or something. 2024 9. It's not here then.
We might just be. I'm looking at recent files, maybe desktop here. Hold on. Is this it? This is it? Okay. Hold on. I need to see, this is the problem when you use a different computer. Hold on. Okay, I think I have this ready. Okay, Chun.
Alright, I know times is short, so I'm trying to be quick. We actually have a lot to talk about. And anyway, this is just my stuff. Oops, well, how do I? Oh, here, okay. So I'm an old guy. I graduated long ago. And I joined USC 4 years ago. I'm teaching 2 courses. Some of you probably suffer in one of these 2 courses, but anyway, my research areas, biostatistics, statistical genetics, and machine learning. I started at Michigan doing statistical genetics, and then moved recently to general biostat statistics, general biostat method development. This is one of my early ones. This is the 3rd cycle now. It got 3 percentile. So the funding should start very soon. These are 4 aims on method development. I'm a little dry here, but basically we try to we have something called CPM, and we try to extend it to random forest and boosting, so that that's applicable in a situation that you have potential interactions among many covariates, and also extend it to penalized version like loss or ridge regression. That's where when you have so many covariate that you no longer afford to put every one of them in your model, so you have to penalize it. And there are some other causal inference stuff. I gave a talk in January, I think, on this one, and now it's got funded. And not yet, but should start very soon. So definitely, if you are interested in working with me on any of these, you're welcome to talk with me. Here is another just a glimpse of what are the stuff behind this grant. So we started this line of research in about 2008. The first paper was published in 2010, and then we said, why not just get a grant out of it? So we started doing that, and then this is now 3rd cycle. One paper that's currently essentially foundation of our research is this one. This was in Aesthetic Medicine, not a very top journal, but it turns out to be a foundation for our research. I gave a talk here about a year ago on this one, and got quite a few colleagues excited, including David, Kelly, and Eric. Basically, this is about something like an in-regression analysis that oftentimes we deal with skewed data, and in biomedical research, there are a lot of skewed data out there. And then we have to worry about transformation. Sometimes people choose log, square root, or whatever you choose. And actually, we can list quite a few ad hoc choices. A more systematic choice would be to go with box Cox. But still, it's a family of transformations indexed by a single parameter. So it's not all possible transformations. It's just a simple family of transformations. But that's about it. And this method basically tried to do essentially a rank-based transformation. So we don't specify transformation, we basically estimate it. It turns out that we can do it. We don't need to specify transformation at all, and we can estimate it along with all the beta coefficients in the regression model. So that turned out to be the foundation for all our recent work, including all those stuff proposed in the grant. We actually spent a whole page describing that method and pros and cons. All right, so that's about it. That's a short way, because if I extend it, it's going to be very long. Anyway, but you can see this major stuff we did in this short slide. This is my other R1. We get 7 percentile, so also the funding should come soon. Unfortunately, I don't budget anything for students. I just budget myself at 10% because this whole grant is so costly. All three PIs get 10% each. But actually, I'm in charge of this whole aim too. We do need to, actually, I budget 50% for year 4 for a student and 100% for year 5. So we do have some for students, but certainly not enough. Basically, we want to do a relatively comprehensive prediction model building using all possible techniques, including certainly so-called AI techniques, that to predict the individual CBC risk. CBC means contralateral breast cancer. So that's for breast cancer patients, they go through radiation therapy, and some of them develop breast cancer on the other breast, and that's called contralateral breast cancer. And so we want to study the effect of radiation and genetic background and all the other risk factors interacting with radiation as the how they collectively confer risk for the other breast of cancer. We will use MAC data here for replication and generalization to confirm our result, whatever result we find. David, MAC data is part of it. This is one other thing. I also collaborate with other people. So I have data accessible for students to get exposed to. For example, this is one I collaborate with Bridget Fernandez at UCLA, CHLA. She studies Hispanic children with autism disorder. We still have maybe 2 more minutes. So we got DoD grant funded, started this year, but no funding for students. Then for 3 years, and an R1 to be submitted. The R1 goes 20 percentile, 23 percentile, I think. So that's quite good, but not fundable. Basically, to study autism Hispanic children in the LA area. Genetics really involved, but my involvement is mostly not genetics, although I do offer advice on that. Because she has a Toronto team to do genetics work. I also collaborate with Andrea Donave to study PCOS, the genetics of it, and specifically we focus on subtyping and try to, because if you do a GWAS on PCOS as whole, you may find something, but subtyping essentially, PCOS, many diseases are like complex diseases. They're essentially etiologically there may be subtypes. Being able to identify subtypes and doing GWAS on subtypes may help you understand better and also identify more genes that work only for specific subtypes. So this is basically our work. All right. So another thing, final plug. So recently, Kelly and I got contacted by a colleague here at PPHS. His name is Berseratinia. He goes by Hamid. So he studies tobacco and vaping, and he says that he wants to offer long-term support for our PhD students to help with this data analysis, especially RNA-seq analysis and omics data analysis, comparing basically 3 cohorts: vaping, smoking, and non-vaping, non-smoking group, to identify genes, consumer patterns that are associated with outcomes relevant to vaping, smoking. So there's no guarantee that there will be method work coming out of it. As a PhD student, your dissertation should be all method work, so no guarantee of it. But oftentimes, when you deal with real data analysis, you get something out of it. You realize there's a need for further development of certain methods for that application. So, if you have interest, contact Kelly or me, so that we can get you connected. That's it.
Quick questions. Ian, here. Right. Not. Thank you to all the speakers. See everybody next week. Thank you.